Let's talk about some of those placebo-controlled trials. First, perhaps with the ones that you guys are running. One can do it from a lab, which I think is what Vince is now planning with Rob and stuff. So can you guys talk about, I'm not sure what the status of that study is. Obviously can you talk about the methods and the goals and any, I don't know if there's any results, or you can give us an update on recruitment or anything?
Sure, I can tell you a bit about our upcoming study. We haven't started recruiting yet, so I can't tell you anything at all about results or anything that we're finding. We'll be recruiting shortly. But what we're trying to do in this study is look at really the immediate effects of microdosing. So this time, not so much focused on changes that result from a long period of microdosing, but really we're trying to see if we can find any cognitive or physiological markers of something like a microdosing state or state-based changes related to microdosing. So we're going to get people who are already microdosing, regular microdoses, to come into the lab. We're not going to administer substances or provide substances. We're just going to ask them to come into the lab on a day that they're microdosing and a day that they're not microdosing. We're going to run a series of brain imaging tasks using MEG. The main sort of aim of that part of the study is to look for changes in resting state and also changes in novelty perception or pattern recognition. That's the sorts of tasks that we're running there. And we've got some other behavioral and cognitive tasks on a similar theme, really looking at whether people's low-level attentional responses to stimuli are different on the microdosing compared to not microdosing days. And as well as that, we're going to look at a series of physiological biomarkers to see if there are any sort of more easily definable objective indicators of what happens when someone microdoses.
What kinds of biomarkers are you looking at?
So we're looking at things such as BDNF, cortisol. We've really got quite a suite of different genetic sort of biomarkers. Maybe Rob could speak a bit more about the rationale behind some of those measures.
The goal really is as was sort of, already highlighted by David and Vince, is that, we're seeing these hints at stress relief, depression reduction, mood lift, etc. And these are things that have known biomarkers already with them. And so the idea is that if there's less of a signal to see in the outward perception, because the whole idea here is that this is a lowly perceptible or not perceptible treatment that then is providing benefit. So really those sorts of biomarkers provide better visibility into what we're actually looking at. And so that's the interest there, is it gives us a better tool to look at this situation.
And you're recruiting from people who are already microdosing, or also trying to find people who have treatment-resistant depression and maybe are just curious to try it or something?
No, this study isn't really looking at clinical effects of microdosing. We're recruiting only healthy controls for this study. A couple of reasons for that, but the main reason is really that is a much easier sort of study to get approved by ethics. I mean, any of this kind of research at this stage is it's a little bit contentious, and there are certainly some interesting back and forwards with ethics committees to make sure that we do everything in a safe way that's really going to care for participants. And so at this early stage, when there is so much that we don't yet know about these kind of immediate state-based effects of microdosing, we're aiming to try and look at these measures in a healthy population, and perhaps down the track move on to seeing if this is more appropriate for people with depression or other mental illness.